Today we're going to cover prokaryotic and eukaryotic cells. This is part of our biology unit. So back in the day, when microscopes were first developed, they were super simple. They were just basically just two lenses, and the lenses would magnify just a little bit. So there are two main scientists that kind of started working with microscopes, and one was Robert Hooke. He came in and he would cut with this little microscope. Microscope has these lenses, and he would cut thin slices of cork. When he would look at those corks, he noticed that there were these little holes or these little openings inside, and they looked like monk cells. These were the rooms that the monks would live. And so he called these little rooms cells. The next one was Anton van Leeuwenhoek, and he looked at pond water. His microscope was different. It was really small. It's just a small little microscope, a little magnifying glass, and he would put pond water in it. And he was able to see all these things floating around in the water, and he thought they were little animals. And they actually called them animalcules, is what he would call these little animals that lived inside the water. Over time, we developed a type of cell theory, and we had Matthias Schleiden. He comes in and he realized that, hey, plants are made of cells. And then you have Theodor Schwann. He comes along and he says animals are made of cells. So they're starting to recognize that cells are big. They're in animals, they're going to be in plants, so they're all over the place. Next you have Virchow. He comes in and he starts pulling what everybody else has been looking at all this time. And he comes up with basically our main three ideas for our modern cell theory. He said that all living things are made up of cells. So anything that is living is made up of cells. Plants are made of cells. Animals are made of cells. He recognized that cells are the basic unit of structure and function in all living things. Anything that is alive is going to have cells, and that's going to be the basic object that functions in your body. And then new cells are going to come from pre-existing cells. There was a time when they thought that cells just magically appeared in what was called spontaneous generation. That if you had opened a canister and it had meat on it, and then you would come back the next day and maggots were on it. They didn't think that the flies were coming in laying eggs and that's where the maggots were coming from. They thought it came from the meat. The meat would all of a sudden spontaneously produce these maggots, or that granaries produced mice, not knowing that the mice came in to eat the feed. So this was starting to really change what people were thinking of with spontaneous generation, and the cells had to come from pre-existing cells. So things that are alive have very specific characteristics. If I went outside and I asked you guys to point out things that were alive, you guys would easily be able to do so. You would point at trees, you point at grass, you point at insects and birds and animals and people, and you would know that they were alive. So they all have very specific characteristics. If I picked up a rock, you would tell me it's not alive, and then I would say, well, why? And you would tell me, well, it doesn't move, it can't breathe, and so it doesn't eat. So all of these things are characteristics we recognize as something that's alive. So all living things that are alive have characteristics. They have to perform some type of chemical reaction. So when we look at plants perform photosynthesis. That's a chemical reaction. Oxygen, carbon dioxide comes in, oxygen gets broken down and released, and so plants are able to perform that chemical reaction. In humans, we do the same thing. We're going to break down oxygen, produce carbon dioxide. We can break down starch. We store glycogen. We can break that down later. So we're able to do all of these types of chemical reactions in our body. We have to be able to evolve and change over time. So for example, the woolly mammoth, if we look back over time, we can see that these have changed and they have evolved into what we consider our current elephants that we have, our African and Asian elephants. They have to be able to respond to stimuli in their environment. So sunflowers are going to respond to the sunlight. Birds are going to respond to their offspring. So there's something in the environment that's going to stimulate them to do certain things. All living things have to be able to reproduce. And there's two different ways. You can do asexual reproduction, which is basically you're making a clone. You are budding. So this here, you see these in some type of marine, like coral pieces, stuff like that. They will produce a bud that is a genetic clone, a genetic copy of the parent, and it will get released. You have sexual reproduction where you have two gametes. You have an egg or a sperm, and then they're going to come together and they're going to combine to make some type of zygote or seed or some type of offspring. But the offspring is going to be a mix of parent genetic material. All living things have to produce homeostasis, and that's a balance. So when you get too hot, you sweat. Your body is trying to get you back down to your set point, your normal temperature. When you get too cold, you're going to shiver, and that's a way of increasing your temperature to get you back up to your set point. So your body is going to do what it can. It's going to open up blood vessels, have you sweat. It's going to narrow blood vessels. It's going to have you shiver. It's going to do whatever it can to try to get you back. If you drink too much water, you're going to have to go to the bathroom. If you eat too much sugar, your body's going to produce insulin to help break it down. So your body is constantly trying to change those fluxes of imbalances in your body. You're all going to have a common ancestry. So if you look at, for example, the woolly mammoths again, those are going to be ones where you're going to see a common ancestry. You see them with horses and with fish. So they're all going to have some type of ancestor, and the further you go back, you can see changes in that line. We have to be able to make and remove waste. So we inhale, we exhale waste. We're going to eat food. We have to break down that food, so we're able to do that with waste. We have to be able to move and grow. So cheetahs are going to be able to run. Plants are going to be able to open up leaves. Open up petals, so they're able to do different things to be able to grow and to be able to move. Cells can be two types. They can be autotrophs. Auto means self and troph means to eat. So they can make their own food. So things that would be an autotroph are going to be plants. You have hetero, which means different, so different foods. They have to be able to find food. So they have to be able to ingest or bring in food. They have to consume other living things to make that possible. So that could be a bear eating a fish, a human eating a hamburger. Those are things, we are all heterotrophs. We have to bring in food to be able to survive. So there's two categories of cells. You have prokaryote and you have eukaryote, and they all have different characteristics. Prokaryotes are very simple. They're very small. They have ribosomes. Ribosomes are one of the only organelles inside of a cell that are not membrane bound. So prokaryotes do not have any membrane bound organelles. They're very tiny, very simple, just basic stuff inside. They do not have an enclosed DNA. They have a type of DNA, but it is not enclosed like a eukaryote. They are mostly found in bacteria or archaea type of cells. Eukaryotes are going to be a lot more complex. There's a lot more stuff inside of a eukaryote cell. They're going to have more membrane-bound organelles. They're going to have ribosomes, but they're going to have organelles that are membrane-bound, which means they're going to have that phospholipid bilayer. When we talk about macromolecules, they're going to have that lipid macro molecule phospholipid that's going to circle those organelles. They're going to be a lot larger than a prokaryote, and their DNA is going to be enclosed or protected. And you're going to find these in animals and plants. Now what they have similar is they're going to have DNA. It's going to be a little different for both, but they're both going to have some type of DNA setup. And they're going to have a cell membrane. So the cell membrane is going to be kind of like a protecting mechanism. Eukaryotes will have a cell membrane. Prokaryotes will have a capsule, and inside that capsule they'll have a cell membrane, but they'll still have one. And then they're both going to have cytoplasm. This is the jelly-like substance that's inside that allows material to move inside the cell or the prokaryote. So here I have a prokaryote. It's a lot smaller than a eukaryote. Very simple. I've got a nucleoid. This is my DNA. It's circular. And so that nucleoid just free floats inside the cytoplasm. Cytoplasm is this jelly. The prokaryote can have a capsule on the outside. I'm going to have a cell wall, and a plasma membrane is here, this part. I can have cilia, I can have pili, I can have a flagellum, I can have all of these other pieces on the outside. And then I have little ribosomes that will float inside. Here I have a eukaryote, a lot bigger, and you can see I've got a lot more going on inside this eukaryote cell than I do with the prokaryote. I've got cytoplasm, lysosome, smooth ER, free ribosome, centrosome. I've got all of these things going on in this. A lot more complex. All of these are going to be membrane bound, those phospholipid layers, except for the ribosomes inside of my eukaryote cell. There's a lot of diversity in cell shape and size. If I'm going to look under a microscope and I look at my hair, or I look at skin, or I look at the cells inside of my mouth, they're all going to look a little different. Blood cells are going to look different. All of these cells are going to have a very specific shape, and they're going to have a very specific function. These are very specialized. Now, what's nice about specialized cells is they're going to be very good at what they do. And they're going to go in and they're going to take care of business when they're in there. But the problem is, if one of these cells stops working, all of a sudden I've just thrown a kink in this machine. If my red blood cells stop working right, what's going to happen to you? It's not going to be good. And so when a specialized cell starts to malfunction or is unable to do its job, it can start to mess up the whole system. So specialized, they work well, they work quick, but when they're not working the way they need to, specialized cells can be a problem. When we have cells and they're not super big, like they're really tiny. We don't have a lot of really huge cells. Eggs are about the biggest cells that we have. But the problem is, why do we not have a lot of huge cells? And a lot of it has to do with cell size. It's limited to its surface area. Surface area is the outside of the cell. To the volume, how much space you're taking up. So it's limited to its surface area to volume ratio. As a cell grows, this volume is going to increase at a faster rate than my surface area. So we do a lab with my AP Bio. And we take cubes, and we take big cubes, medium cubes, and small cubes, and we dip them in like a bleach solution. And then when we stick them in that bleach solution, you're going to have a layer where the bleach is going to be able to come in. But as I get smaller, what you notice is the bleach is able to get in further. And then same thing here, the smaller ones, the bleach is able to get in a lot further. When I have cells that are really big, I'm not able to get as much material in. It's going to take a long time to get all this material in to where it needs to go, do what it needs to, and then get all the way where it needs to go to get out. It's a lot of traveling for that. Whereas if it's smaller, I can move in faster. I still got to come in, come out. But when I have a small cell, I can move in quick. I can get out quick. I'm using less energy to get out with a smaller volume. If I have more surface area or smaller volume, it's more efficient. So when we talk about cell size, this is why cells are not big. I have too much volume, and that volume is inefficient for that cell. The smaller my volume is, the more surface area I have, the smaller it is, the easier it is. When we look at an organ like villi in your intestines, they're like little fingers. And so I have all of this surface area for my villi, and food comes in and gets in between there and absorbs out. So I have a lot of surface area and low volume. This is very efficient at absorbing stuff very, very quickly. Where when I have something huge like this, it's going to take a long time, and by that time the cell would die. It's not going to be able to function. All right, that's it. Prokaryotes, eukaryotic overview. If you guys have any questions, let me know. Talk to you later. Bye.